getting ready to start DNA extractions, and we're actually working on that now. First step of DNA extractions is understand that the chicken has enucleated blood, so you want to start right here where it says 1B. Very simple. Pipette 20 microliters of protein kinase K. So that's what we're doing right here. We're taking our pipette and pipetting in 20. This is our protein kinase K, and I'm going to be okay, and then place it in the tube. Okay, that's our first step. K into here, 20 microliters. Then we have to put in 10 microliters of the blood. So we basically got our tubes labeled, and this is actually kind of difficult because the blood is actually a little bit thick, and you want to pipette. Now, the actual part of getting in there and getting the blood is try to mix it around so you get a lot of blood cells. And a lot of times, until you actually come up to the top, it won't even pull it in. It's that thick.
Wow.
So there is some blood in there, but we're going to go down a little lower, see if we can get more cells, darker, redder cells. There's some right there. We'll take that, and then we'll pipette this into the protein kinase K. And sometimes it's a little hard to get it to let go, but there we go. All right, now it's in there. Now we're going to do the other 6 just like that and turn it off.
Okay.
Go ahead and do this. This is my buddy, Ashley Payne. Hi, everybody.
Hey.
And go ahead and pipette this blood. And so the samples will have different thickness, different consistencies, and you just have to
Whoa,
yeah,
that's so thick.
yeah, give it a little time too, and spin it around, mix it, make sure you get some good old red blood cells in there, and then try it, see if you can get some. Okay? Okay, go real slow. No, real slow. You got some. Go ahead. This is your 915. See if you can get it down into the protein kinase. That's what's going to break it all up.
Okay.
Try it again. Keep going. Okay. All right.
Okay.
You did it. Now. Okay.
I'm recording.
Okay. The next step is to take this 1X PBS. What the directions are going to tell you is to take pipette the extra volume up to 222, okay?
Mm-hmm.
But we've already put 30, a volume of 30, when we added the protein kinase K and the blood, okay? So it's now up to 30 microliters. So we're going to top it off with 180 of 1X PBS.
Mm-hmm.
Okay, you can. And let's go.
Okay.
And then here we go. So each one of the samples is going to get 180 of PBS. There we go. And then the next step is going to be to add 200. So you would set, here you go, you would set your pipette on 200. Step is going to be the buffer AL. You want to add 200 of buffer AL into each one of your samples. So we're going to get another pipette tip and then open up our buffer AL and put 200 into each one of these. Okay. Okay, you can turn it off now.
Okay.
Slice buffer in, and we've got the protein kinase. What we have to actually do now is vortex and mix and then place on heat, 56 degrees for 10 minutes. So we come over here, we put our samples right here, and we vortex them and mix them so the lyse buffer can open cells up and we can get this DNA extracted. So that's pretty good mixing. And then we're going to put these into and then mix this last and final one. Okay, now come with me, and I'll show you where the heater is. We have a heater over here that is set at 56 degrees. Yeah, I've already set it. So, and you can see by the thermometer.
And the heater.
Okay, starting the heater. Now we get our timer. We get our timer right here set to 10 minutes. Okay, and we push start.
Okay.
And then we set that there. When that beeper goes off, we pull our samples, and then we add the ethanol to clean it. We add 200 of ethanol to clean it.
Okay.
So anyway, and that's the good of it. Thank you very much. We'll go back to the next step here in a minute whenever we get done with our 10 minutes.
Okay.
I don't think I meant to do that. That's okay. I meant to take a picture, and it was...
You can see the timer is almost done. We're down to 21 seconds. And Ashley is on standby with his beloved pipette. Da-da-da-da-da-da-da-da-da-da, super scientist in action. Okay, grab your 100% ethanol. And you can open that lid up and set it somewhere where it won't flip over. Okay, now we turn off our timer. It's been sitting on here for 10 minutes at 56 degrees Celsius. We pull out our samples and we put them over here. Put them over here. And what we're going to do with the ethanol, what the ethanol does is the ethanol cleans it. So, here we go. Ashley, I'm going to open a few of these and you pipette in the 200 that they need in each one of them, okay?
Okay.
Okay. No, yeah, there you go. Make sure it all goes in there. All right. You don't have to go all the way down with the ethanol. With ethanol, we pipette into each sample.
Mm-hmm.
And there we go. I didn't get it. Okay. Go ahead and put that off.
Okay.
There. After you've added the 200 of ethanol, you need to vortex it again. It's kind of like a cleaning process for it. So come back over here to our vortexer. Get a close-up of our lovely vortexer. Come on over here. This is our lovely vortexer, and this is how we vortex. We're vortexing it right up, getting all that good stuff mixed up, and then we're going to place these in filters. Filters that will hold the DNA in it, and usually it's much cleaner DNA when you use filters. I'll show you that part in a minute. Okay? So, go ahead and flip off. Next step is going to be to take your sample and pipette it into this spin column. This spin column right here is what actually separates the DNA and the fluids that we use to clean it. And just basically make sure everything's marked correctly, and okay, just till all your fluid is over there. And there we got it all. Hallelujah. Okay. Now you can flip off.
Okay.
Now, what you're going to have to do is centrifuge. Okay, this is our centrifuge. You have to centrifuge 8,000 RPMs for 1 minute. So I'm going to set this on 8,000. Okay. And then I'm going to centrifuge, and I'm going to, when you use the centrifuge, you have to make both sides equal weight. All of these weigh the same. So I'm going to put 3 over here and 3 over here. And the important thing to know is that I have a marker here on this one and on this side. You have to realize that these are going to be exactly opposite of each other, and that it is completely balanced. If not, it won't work. So we're going to put this in, shut the hatch, and push start. And let that centrifuge for 1 minute. When it's done centrifuging, what you have to do is, this is the part where you should be very careful. Just take out the top. You don't want any of the fluid touching this filter again. So you take out your sample and you place it in a new one. Okay, then you just discard this old one. You don't need it anymore. We're going to do that with all the samples. We have 6. So here we go. And let's see. Okay, and then the next step we will place in 500 AU of buffer AWL. Okay, here's this, the buffer AWL, right here. So Ashley, you want to do that for me, dear?
Yep.
Go ahead, Ashley. Pipette 500. Be real careful. Into every one of these. I'm going to open the top for you. This is the AWL buffer. You don't want to touch the bottom.
Okay.
Okay. All right. There's one. Okay. Can you just stop for a second? Touch anything. Yeah. Be real careful not to contaminate. Okay. Okay, hold on. Go ahead and put that back in there. There we go. After we have pipetted the AW1 buffer, 500 microliters is what we use into each one of the samples. We're going to take them back to the centrifuge and start them at the same thing for 1 minute. It's a spin for 1 minute and at 8,000 RPMs. So there we go. We got that. Then we put this on here like this. We shut this hat and then we hit start. And it'll run. We're done with our minute. We're going to open. Now, one thing, again, is we don't want the fluid to touch the filter. We're going to put them in whole new catchers, in each one of them without the fluid going back into the filter. That's very important. So, don't want to contaminate your DNA or anything like that. So, you just basically throw the old container away. And then after this, we need to pipette 500 of AW2 buffer, which is this buffer right here. AW2 is the next step, 500. So go ahead and put that off. I'll let you do that, Ashley.
Okay.
Very technical, delicate thing, this pipetting and getting it correct. So there you go. All right, I'll shut that for you. All right. All right. All right. And then the very last one. All right. Okay, now we go on. Okay, now that we have put in 500 of the buffer, the AW2 buffer, we need to spin for 3 minutes because we need to dry these out. So we're going to come over here, place them back in the centrifuge once again, make sure everything's aligned. And over here, and then close this up. And then down here is where we change everything. We change it to 3 minutes this time, because this is our final step before we catch our DNA. And then hit start. All right. This is kind of a technique that I have to do kind of different in my lab it's going to tell you to pipette 200 microliters of buffer AE. This is the final step, okay? It's your buffer AE. But the important thing is I need a pretty high concentration of DNA. Mm-hmm. So what I do is I cut it into, I run 100 microliters through the filter, and then I take that out, and then I run another 100 through to make sure I can get my highest yield, or a higher yield than what it's going to give you if you run the full 200 through at once. So we're trying to get a higher concentration of DNA. That's why we do this. So,
okay.
So now after it's centrifuged for 3 minutes with the AW buffer, you want to open it up. Open it up, and here we go. Mm-hmm. And what I'm going to do now is kind of show you how to actually put it. Once again, you don't want the fluid to touch anything, to go back up on that filter. That's bad. So we come over here and we place our sample into a new tube, a catch tube. These are the 1500 microliter tubes. And then you open right here. And the important thing about this is when you pipette your AE buffer, you don't want to touch the sides. You don't want to touch any of the sides of the tube because it has contamination on it. The only thing you want to get is the sides of this tube. You do not want to touch. You just want the fluid to fall directly on the white filter. That is so important that it just fall on the white filter. And okay. And that's how it's done. Do your best. It doesn't always happen that way, but that's the way you want it. You don't want it to touch the sides of the tube if you can at all help it. Mm-hmm. Okay, let me turn off that. Okay. Okay, trying not to touch the sides. You did pretty good there, my friend. Okay. All right, go ahead and close it up. And then the last sample. And then what we do is we let them set for 1 minute to 2 minutes. We let the fluid set because it's got to loosen that DNA off the filter. What number you got there?
7.
Okay, so we need number 7. There we go.
Yeah, okay.
So go ahead and try that one more time. Once we give it about 2 minutes to sit there, then we spin it for 1 minute, and then we do the same thing over. We do the same exact process over, and we extract. About my centrifuge is pretty powerful. So whenever you're placing these, you notice you have an open lid because you've got a catch container now, and you've got a closed lid. When you put them in here to be centrifuged again, make sure you put these lids down here at the bottom. If you don't, it kind of throws it off centrifuge balance, and then on top of that, it'll break the lids because it moves that fast. It really does. So, and I think we're going to have to adjust our speed on the centrifuge. So,
okay.
Grab this. Okay. Grab this, and now we're at 60 Gs, which is 8,000 RPMs. So we're going to shut it up, and then we're going to turn this on. I'm done centrifuging. None of my lids broke, but if you would have turned the lids upward like that, they would have broken. They just, I don't know, the centrifuge itself has a G-force that'll just knock these lids right off if they're not placed in there correctly. We now have our DNA. We have to do something. This is our product, our DNA. And we have to find out the concentration and how much we got. We use this thing. Come on over here. It's called a nanodrop. Nanodrop to figure out our DNA concentrations. Okay? And basically it's used with this computer. And we're going to do that right now. So. All right, this is the last thing we do. We take our DNA, got a shot of the DNA,
Mhm.
we open it,
Mm.
and we get a pipette tip, and this is called nanospecking, and it'll let us know how much DNA we actually get. So then we wipe this down with a Kimi wipe, and then go into it. We get 2 microliters of DNA. 2 microliters of DNA. We place it on the nanospec. See the little drop there on the nanospec? It's got to be right on there. And then we close this down, and then the computer will tell us how much we have. Okay?
And that is how you extract DNA. Later on, we'll go over how to extract microRNA and RNA. And that will be 2 similar processes, but some different steps. Anyway, have a good day. Bless you.